Okay. Welcome everyone to our first journal club for this year. I'm actually very happy to have our guest today, one of my former colleagues. Um, so he's going to talk about his class paper on fNIRS and rate variability during self-compassionate thinking related to stressful memories. This journal club is provided by NIRx, so we are a company that's always trying to innovate and be very together with the researchers. So that's if you have any papers that you want to see here, any cool science that you're seeing out there, or your own paper, please let us know. We're very happy to hear about your research and have you here at the Journal Club. So the webinar is then moderated by Amy, Alina in the back, and I will be hosting today. Some considerations, so all of you are muted. Questions are welcome at any time. Please use that Zoom chat. The content will be available in our website in the upcoming days. And if you have any further questions, please don't hesitate sending us an email at consulting@nirx.net. Hey, hope you enjoy today. So I will introduce our speaker, Paulo Bazán. He has a Bachelor of Ciências Moleculares from the University of São Paulo. He is currently a research assistant at Instituto do Cérebro at Hospital Israelita Albert Einstein. He is also a PhD student at the Radiology Department of the Medicine University of São Paulo, and also has a PhD research project focused in fNIRS and hyperscanning setup. He also participates in interdisciplinary research projects and biomedical investigation, laboratory focused on neuroimage and cognition. And Bazán is also very used to help a lot of researchers in Brazil. So we are very happy to have you here, Bazán. Please, floor is yours.
Thank you for having me. So let me share my presentation. Just 1 second. I guess it's okay. Now you can see it, right?
Yep.
Good. So first, let me start giving a disclosure statement. As Made mentioned, I am a research assistant at Albert Einstein and also a PhD student in INRAD at the Faculdade de Medicina of USP. But I'm also a freelance scientific consultant, and I've, among other jobs, worked with NIRx and brain support in the past. So that's what Made mentioned about helping other researchers. Okay, so today we are going to talk about this article from our group, talking about fNIRS activation and heart rate variability during self-compassionate thinking and its relation to stressful memories. This is a work from Fabio. It was his postdoc work, and I helped him during this project, mainly mostly in the analysis part of this project. And Maria Delia also participated in this project as well. So let's start talking a bit about self-compassionate thinking. I guess compassion is more usual concept that we know, that we think about others and have compassion towards others. But we can also have compassion towards ourselves. So in this sense, self-compassion would be really similar to compassion, but focusing on ourselves. So it also involves being touched and opened to our own suffering, and in a way that we are not trying to disengage or neglect our suffering, but we are trying to deal with it and be kind during this deal with our suffering. So one important aspect is that it's a non-judgmental understanding of one on pain and failures and inadequacies. So that's the general idea of self-compassion. It's basically the same concept of compassion, but focusing on ourselves, on our experience. In order to give an example of this is a good example from Svensson. We can separate self-compassion in three aspects. So self-kindness, common humanity, and mindfulness. So we can compare like what would be self-kindness against self-judgment. So the ability to treat oneself kindly with compassion, or we can criticize and keep remembering our failures and thinking, oh, that's not right. So that would be the opposed effect of self-kindness. The other aspect is the humanity aspect. So our suffering is part of human experience. As a human being, suffering is part of life. And when you realize that, you have this common humanity sense, instead of isolating yourself and thinking that's something that's mainly related to you, and so you isolate from the others, people in your life, and you think that's your problem only. And being mindful of that experience. So being aware of what you're going through, but without any judgment. So you are conscious, you are aware of this experience that you're in pain or in suffering, but when you think about it, you don't really focus on over identifying the negative feelings. So this would be the contrasts between self-kindness, self-compassionate thinking, and the opposite way. Self-compassionate thinking can be seen as a form of emotional regulation. And emotional regulation, if we look at the brain areas involved in emotional regulation, there are several areas. Mostly when we talk about emotion, we think about subcortical regions like the amygdala, mainly that's related to the processing negative emotions. But we have other cortical regions important, especially for regulation of these emotions, not only the initial processing, but for regulation of these emotions. That involves the dorsolateral prefrontal cortex, ventrolateral prefrontal cortex, the anterior cingulate cortex, and in general, the prefrontal cortex, apart from the amygdala, insula, and other subcortical regions. There are several models of this emotional regulation. It can be more explicit regulation or implicit regulation, and it can be more based on previous experiences and like model-based regulation when you have more exposure towards this stimulus and you are more trained to deal with them, or more innate and based on error prediction and more reactivity regulation. So these areas related to these kinds of regulation vary a little bit, but in general, they involve the same system. It's the anterior cingulate cortex, amygdala, ventromedial prefrontal cortex, mainly. But there are several types of emotional regulation. When we talk about self-compassionate thinking, it can be seen as a reappraisal emotional regulation technique. That's one specific form of emotional regulation. In reappraisal, you try to give new significant, new values to that experience. So when you think about it, you change the focus of what you're feeling, what you're having during that experience. And related that to prefrontal cortex activity, this articles is one example. There are other articles in the literature studying this effect, but these prefrontal areas are involved, especially in this reappraisal technique. So dorsolateral prefrontal cortex, orbital frontal cortex can be seen and activated when we compare stimuli when we are trying to apply this reappraisal technique and when we're exposed to the stimuli without any techniques, just observing, attending to that stimuli. So basically this article, this image is an fMRI study comparing these conditions where the same images, same stimuli is presented, but in one case, the instruction is to use this reappraisal technique. Okay. This just show this prefrontal cortex region involvement in this activity. And there's another part of the system that is the integration with the visceral part of our system. So this prefrontal cortex activity regulates the activity in cingulate cortex, and actually it's regulated in both directions. So cingulate cortex communicates to prefrontal cortex, to amygdala, to insula. They're all entangled together. And in general, when we have more prefrontal cortex activity, we have less sympathetic activation and more parasympathetic effect. And that would reduce the heart rate. In the opposite direction, we can have lower prefrontal cortex activity, which would represent higher sympathetic activation and higher heart rate. This is mediated from this communication from prefrontal cortex, passing through the hypothalamus, brainstem, and then going down to reach the heart rate connection, okay, so heart connections. When we look at heart signal, we have some signals that are established in the literature that reflect this relation between sympathetic and parasympathetic activity. So we can use a few parameters, a few measures of heart rate to study these effects. One of them is simply the difference or the variation, the standard deviation between the peaks in the R peaks. This is called the standard deviation of normal to normal peaks, because it's basically the R peak, but correcting for artifacts or incorrect R peaks related to other signal. So the normal only means that it was pre-processed. So this standard deviation of normal to normal peaks is informative of this activity of balance between parasympathetic and sympathetic activity. Basically, in general, we're talking about the previous slide about high heart rate and low heart rate. But when we have higher heart rate, the variability of this heart rate decreases. And when we have low heart rate, the variability of these intervals increases. So the higher variability of these intervals would represent more parasympathetic activity and related to more prefrontal cortex activity. Okay. Another measure we can have is the frequency, separating this heartbeat signal in a power spectrum and trying to evaluate different frequencies. We chose here in this article to highlight the high frequency spectrum because it shows a signal that's not that similar to the variability signal. Both are related, but the heart rate variability that I mentioned in the previous slides is more related to these lower frequencies. So this represents a bit of another aspect, mainly related to the respiration effects and associated with parasympathetic activity. So I mentioned about a bit about what we see in the literature. Now I introduce these object studies, the study objects. So we decided to evaluate this prefrontal cortex activity and heart rate variability during a more ecological self-compassionate thinking. We wanted to try to give this experience applying this self-compassionate thinking that's not so much common in the literature, and trying to relate these parameters that we can observe of prefrontal activity, heart rate variability and correlate them, and in healthy subjects. So we wanted to have a starting point for then trying to evaluate patients. But this would be suggestive our model would be good to study this effect. So we propose a new task basically to try to see this more ecological effect. We have the hypothesis based on the literature that I mentioned that self-compassionate thinking after exposure or recollection of stressful autobiographical memories would decrease the level of cortical activity. And that the self-compassionate thinking associated with these autobiographical memories would increase this heart rate variability. And I'll mention a bit about this decrease of cortical activity. In the previous slides, I mentioned more higher activity during the emotional regulation. But the problem is that even though we didn't apply any technique during the stressful memories, a natural effect of prefrontal cortex activity to deal with that is associated. So our hypothesis of decrease during this special self-compassionate thinking would be because we would have a less arousal, less other aspects of the system of regulation that would be involved in this task. But we would still have the increased heart rate signals expected. And we also expected to see that's a relation between prefrontal cortex activity and the parameters, all right, in some specific regions of this prefrontal cortex activity. So here's what we did, what we tried to do to evaluate these effects. We have two different runs, two different tasks running sequence. Because first, we tried to engage this stressful autobiographical memory condition in which participants were exposed to an audio, guiding them to think about the stressful memories, alternating with a control condition. And then we have another block design task in which the audio guided them to now apply this self-compassionate thinking, alternating with the control condition. It's important that we did always first the stressful autobiographical memories and then the run of self-compassionate thinking, because we needed that the participants to have this experience of stressful autobiographical memories, then to be able to apply the self-compassionate thinking. So this is a limitation of the general paradigm, not that we cannot change the order of these runs, because we want to apply the self-compassionate thinking after this autobiographical memories recall. But maybe in future studies, we can try to think a different clever way of studying this. And both runs have the same duration, so 350 seconds, almost 6 minutes. And during these tasks, the participants were having their fNIRS signal measured and their heart rate measures. The fNIRS signal was measured with the NIRS-Port1 system. In this case, we did not use short distance detectors. We didn't have them at the time. And this heart rate recording was done using polar system. Just to give an example here of the audio the participants were hearing during these tasks, the stressful memories would try to bring these memories and trying to make them experience the feelings associated with these memories. So really try to remember the details of the situation, the pain, the frustration. And this audio was basically these sentences. Now try to bring your mind a recent situation of suffering. Try to remember the details of the suffering. whether you felt pain, frustration, or what other feelings. Then bring those memories to your mind. Remember how you felt. So the sentence were repeated to bring those situations. After that, in the control condition in both tests, the instruction was to take a break. The audio instruction was to take a break. So the instruction was, let's take a break, stay in your natural state, try to disengage from any task, stay alert with your eyes closed, let your thoughts flow without thinking about anything. So one important thing to mention, participants were always with the eyes closed to try to really experience those memories, right? And then we have the instruction from the self-compassionate thinking condition that would be repeat internally by trying to fill each sentence. May I accept my suffering with kindness? May I recognize that suffering is part of life? May I be kind and loving to myself? May I be free from pain and suffering? May I be at peace and be happy? So it was simple sentences. It's important that here we didn't do like complete training of self-compassion, right? That's a different thing. So it was just this brief instruction to apply this self-compassion at the moment. And here are the regions we measured with fNIRS. We mainly focused on prefrontal cortex, in both hemispheres. So the image shows the 2D montage, and the table is based on the main measured area, main area that provided signal based on the models from Fold software. That's a software used to estimate the reconstruction of the image and trying to check which brain regions would be measured based on the channel positions. Okay, so mainly we checked Brodmann areas in the frontal regions, Brodmann area 46, 9, 10, and that's mainly comprised the dorsolateral prefrontal cortex or the frontal polar cortex. In this study, we included 33 healthy adults. 23 of them were female that signed the informed consent for these studies. And the mean age was 33 years old. So let's jump to the results. First, we tried to do an average map of these results of what was going on during the stressful autobiographical memories and during self-compassionate thinking. So in both conditions, we have some prefrontal cortex activity that was expected, but during stressful autobiographical memories, we have more activation, more evident activation in these prefrontal regions. And the effects were especially observed in the oxyhemoglobin signal, but we found coherent signal between oxy and desoxyhemoglobin. And after this serves to understand what's generally going on during each of these conditions. But we also compare them directly. So when we compare these two conditions, we see that we have more signal, more activity during stressful autobiographical memories in general. And here I'll mix results and discussion. I guess this will make it more interesting. So the idea behind this is that when I was talking about emotional regulation, is that we actually are always doing this emotional regulation when exposed or thinking about these memories, autobiographical memories. So even though they had no previous instruction to try to regulate their emotion, only having this emotion already involves this prefrontal cortex regions to regulate these emotions. And the dorsolateral prefrontal cortex regions are really relevant for this regulation, and there's mixed results into the literature relating them more to negative stimuli or to specific regulation processes. So it's something in debate still right now. We know that it's an important aspect, but it's something in debate. And there are studies that try to evaluate differences in, for example, valence of the stimuli and this regulation, or the arousal raised by this stressful condition. And there seems to be difference between more medial region, prefrontal cortex regions, and more dorsolateral regions, regarding one more related to the valence of the stimuli and the other more to the arousal of this condition. So in the case that we use this more negative autobiographical memories, stressful memories, part of the higher activation for this, it's based on this high intensity of the experience, right? That self-compassionate thinking would decrease because you are actually being able to deal with that emotion, experience a less intense emotion. And that would be related to this expected lower signal prefrontal cortex activity during self-compassion. On the other hand, we would still expect this prefrontal cortex activity as we observed in the general, the previous slide, because this prefrontal cortex regions also is important for this relations to the heart rate regulation, right? So even though we have less prefrontal cortex activation related to these other aspects of the stressful experience, we still have this regulatory activation of prefrontal cortex. So that's the basic idea behind the study and how we interpreted these results, and they are in line with what we expected. The other aspect of this study was trying to evaluate the heart rate variation, right? So when we compare basically three values regarding heart rate signal, so the raw heart rate and the two other measures of the standard deviation of normal to normal intervals, and the heart rate, high frequency of this heart rate signal. The significant difference between the stressful autobiographical memories condition and this self-compassionate thinking condition was observed during this with this metric of standard deviation, in which we found the higher variability expected in the self-compassionate condition. And although the other conditions did not reach significance in the tests, they were in the same direction that we would expect. So in the absolute value, the higher frequency signal was higher in self-compassionate thinking, and the heart rate was lower. So all these results kind of agree. But as I mentioned earlier, these metrics have a small differences of what they represent, what they mean. So SDNN may reflect better this balance between parasympathetic and sympathetic activity during this self-compassionate thinking. Another important thing that we wanted to look is the relation, the association between cortical activity and these parameters of variation. So we try to correlate standard deviation of normal to normal peaks and the higher frequency signals of heart rate with the stressful autobiographical memories and with the self-compassionate thinking. So during each of these conditions, if we could see some relation of these signals. Curiously, we only found significant relations, associations during the stressful autobiographical memories, and not during self-compassionate thinking. This was not exactly what we expected at first, but this might be associated with this intrinsic response and regulation required during a more stressful situation. So we only observe this in this stressful autobiographical memories. Other possibilities for this is I feel the limitation of the studies, maybe the sample size, or that we didn't give a thorough training of self-compassionate thinking. It was a brief instruction. So there might have been variabilities of how to apply this self-compassionate thinking and how really, what really each participant was experienced and what they are trying to do during these tasks. So that might be related to not finding this association during this task. Other curious result is that these were more evident in the high frequency heart rate signal, which was not significant in the comparison between conditions. So this might reflect a bit of different relations between this parasympathetic sympathetic balance, more reflected by this standard deviation or normal to normal peaks, or the higher frequency more related to parasympathetic activity exclusively. So maybe that's another explanation why we found this prefrontal cortex activation related to the high frequency signal, especially in these cases. And well, that's the general idea. I guess the limitation of the studies I mentioned a bit is that sample size we did this very brief training, brief instruction that was actually intentional because we wanted to have this more ecological situation with more quick application of this test, of this self-compassionate instruction, to see if even brief explanation like this would have a positive effect and would have an impact on these physiological measures, which apparently it did, which is very interesting. But mainly trying more thorough explanation of self-compassionate thinking and other controls of using questionnaires would be really helpful to further understand these effects and complement this studies. And the other aspect is that even though the task, the new proposed task is very interesting and seems to be adequate to study this effect, we have a few issues when we compare NIRS, the signals from two different runs when using fNIRS. It would be better if we could think about a task that would involve both conditions in the same run. That would provide a better comparison between the fNIRS signals. So that's something that we're still thinking about. For now, as I mentioned, the idea of using this design was based on the necessity of first experience the stressful autobiographical memories to then apply the self-compassionate thinking. But it's something to think about, and maybe other studies, other groups can come up with ideas to deal with that. Okay, so here's the group that worked in this project. So Fabio was the main author, Elisa, the last picture, was the PI of this study. And here's our group at Albert Einstein. This is a photo right before the pandemic, for 2020 here in Brazil. So it was the last time that we have all this group together like this. But yeah, this is our group, and we are currently having a new facilities and trying to expand our research institute. So thank you very much for your attention, and I'm open to questions now. And feel free to write to my email if you want.
Thank you, Bazán. Was really nice. Definitely one of the most challenging design of experiments for this one. You can go back to the gallery view, I guess, if Bazán is still pinned there. Okay, so for the questions that we have, if you have some questions right now, please feel free. You can unmute yourself if you want to make the question directly to Bazán, or you can write on the chat. We do have some questions that came before, and I will ask them. So one of them was about the infant rate variability, actually. So in this case, you're doing with the adults, but is there any reason why or any difference for the infant rate variability?
Actually, one important previous aspect of that in infants, it's how to evaluate and how to train them for self-compassion. It's easier to train them for compassion in general, but compassionate and social interaction aspects usually require a lot of prefrontal cortex activity and integration between prefrontal and parietal cortex activity. So these are areas that are in development during our whole life, basically, but mainly in infancy and adolescence. So few studies have indicated that children do not have full capacity of these more abstract social interactions. They are developing them, and they have a lot of techniques for that, but we have to adapt this task first, thinking about this self-compassion and compassionate thinking. We are actually considering trying to measure this heart rate variability in infants in a project that we are proposing right now. But we are trying to adapt this idea of self-compassion to make it much more objective and more easy to apply for children. So, for example, instead of trying to make more abstract stressful memory and then a more abstract self-compassionate thinking, try to give more object examples for these instructions, and maybe not focus on only self-compassion, but in compassion in general, a bit more feasible for these kids and more related to their experiences. And regarding the heart rate variability, I'm not an expert on it, but we believe that we would find similar results. We could apply this technique to find similar results in children, but that's something that we have to study a bit more and see if these same metrics are appropriate with these kids, because there are other metrics that might be more appropriate. There are several heart rate variability metrics that we could use. And actually there are different timings as well. So for example, here we use 5 minutes recordings because I didn't highlight this, but during this almost 6 minutes task, we selected 5 minutes in the middle of this task for heart rate analysis, because the traditional timing window for this SDNN analysis is 5 minutes or 24 hours. And they reflect a bit of differences, different balance between parasympathetic and sympathetic results. But so we chose this 5 minutes window. We'd have to check if for children this metric is the most adequate. So that's general idea that I can mention right now. And there are several studies trying to evaluate self-compassion in adolescents. So in really young children, it's harder, but in adolescents, there are a few studies trying to evaluate this. And actually there's a questionnaire, for example, that was proposed for adolescents to evaluate self-compassion. So that's something there's some articles in the literature. If you want, you can send me an email and we can exchange these articles. Yeah.
Okay, thank you. Yeah, I think one of the most challenging things on this area, usually well-being, is really to try to make it more objective and less dependent on your self-experience, I would say. That's to really try to be more reproducible and more reliable, I would say. So that's one of the challenges for the experimental design that you mentioned sometimes, like how to really do this, how to you really evoke those emotions and evoke the self-compassion thinking in a very scientific way. So I think everyone in this area definitely thinking about that and trying to bring this to a more scientific approach, I would say. But yeah, very interesting. Okay, anyone else have more questions? I guess. No, okay. I guess that's it, Bazán. There's no more questions here, and we are happy to have you. Thank you very much for explaining your study and coming here today. Thank you.
Thank you for the invitation. It was really nice presenting. And if eventually you have further questions, you can send them to my email.
Perfect. Thank you very much. And if you have any papers or any interesting studies that you'd like to see in the Journal Club, please let us know.